Sorry about being late. Had a lot of emails to deal with and a lot of student emergencies, so that's why I'm late. Just a few minutes, though. My apologies. Let's begin, or actually, there were two questions that I wasn't able to answer last time. Let's begin with that. One of the questions I remember, and that is, were aquaporins, are they found on bacteria? And aquaporins are channels that bring in water to cells, and aquaporins are found on bacteria and archaea. However, they are not found as often as they are found in eukaryote cells. So eukaryotes have more, or at least more species have aquaporins than bacteria, which have more species that have aquaporins than archaea. But all three of the domains have aquaporins. The other question was, are slime layers more often found on gram-positive or gram-negative cells? And the information that I found no difference between the presence of a slime layer on a gram-negative or a gram-positive cell. So they're found on both and fairly similar numbers. Most cells do not have a slime layer, and most cells do not have a capsule or a slime layer. It's only some species that produce the capsule or slime layer. All right, any other questions? Let me state, I did put it in the discussion that there was agreement in the discussion for taking the quizzes 11:59 p.m. Tuesday night to 11:59 p.m. Wednesday night. So future quizzes will be moved to that time. I have not done that yet, and it'll take me a little while to do that, but I will get that done before you take your next quiz. Any questions? If not, let's begin. And I don't think I'm sharing my screen yet, but that's okay. I don't have anything up for you guys to see yet. Can somebody confirm that you're hearing me, by the way? I forgot to ask that. Oh, that's not right. Don't want that. Why is that there? Don't know. Doesn't matter. Hopefully you guys didn't see that. It's fine. It's me in a video recording, and I don't know why that's even here. Can somebody please confirm that you're hearing me? I'm not sure. Maybe I shut the...
We can hear you.
Thank you. Shut the recording down because I had a recording here and I think it was playing and shut it down. All right. Today is May 4th. Let me share my screen now. Come on. All right. Today is May 4th and gosh, why is that there? That doesn't look like that's the right one. Yeah, that's winter term. Why is that here? Oh, I clicked on spring and it opened winter folder instead. Let's get that up here. And we're supposed to be working on chapter 5 and hopefully finish it. We'll probably fall behind schedule and actually start chapter 6 today. I think we're a little bit behind schedule, but hopefully we'll at least finish chapter 5. There is a lab today, lab 7, and it'll be at the normal time. I have not sent out the unknown number, and I'm working on it, and hopefully I'll get it sent out after the lab today. And we're gonna, I did start the gelatin hydrolysis test for you on Tuesday, so hopefully I can get that result sent out next Tuesday. This coming Tuesday, in fact. All right, any questions about what we're doing? All right, if not, let's move on to chapter 5. So the last slide I talked about was factors that can influence enzyme activity, talking about feedback inhibition. Let's mention that ribozymes are a non-protein enzyme. They are an RNA enzyme, meaning an enzyme made up of RNA. The RNA molecule, which is a ribozyme, tends to cut and splice RNA or another molecule, and they function as enzymes in that they have an active site that actually binds to the substrate, forms a complex. They are not used up and modified in a chemical reaction. And then the product falls off, and then the RNA enzyme can then bind to another substrate and make a product out of it. So they are enzymes. They're just not made up of protein. They're made up of RNA. We're not going to discuss any RNA enzymes except for one. And Tutora doesn't mention it, but I will. And that is when we're talking about a growing protein, meaning a growing polypeptide, and when you finish making it on the ribosome, it will be a protein. The removal of the amino acids and the attachment of the amino acids to the newly added amino acid, that is actually done by an RNA enzyme. It's part of the ribosome that does that, and it's an RNA part of the ribosome. So this is one and the only ribozyme that we will discuss. Any questions about any of that? All right, if not, let's change gears and talk about the metabolic diversity among organisms. Organisms have a large amount of diversity, especially microbes. And all microbes require two things. They all require an energy source, and they all require a carbon source. And this is true of all microorganisms or microbes. Do I mean viruses? Yes. But I really am talking about microorganisms, meaning a cellular-based organism. I don't think prions, for example, they don't really require an energy source, and they don't require a carbon source, and they are a microbe. And viruses, I suppose they get their carbon source and their energy source from the cell that they infect. So I guess viruses do. Metabolic diversity can be set up by basing an organism's, meaning we can classify an organism on where it gets its energy source. Phototrophs are organisms. I'm talking about cellular-based organisms. They derive their energy from light. A chemotroph is an organism that derives its energy from an inorganic or an organic chemical compound, such as glucose. And as you know, we are a chemotroph, and we derive our energy source from molecules like glucose. Sorry, I've got a frog in my throat. Green plants, as you know, are phototrophs, and they derive their energy from light. And hopefully you know there are green bacteria, which are phototrophs, and they derive their energy from light. We can also classify organisms based upon where they get their carbon source. And organisms can be classified as an autotroph if they get their carbon source from CO2, either in the air or in the water. And I think most of you know green plants get their CO2 from, well, they get the carbon from CO2. Sorry, didn't word that right. Any question about that? All right, there are other organisms, and they get their carbon source from an organic chemical molecule, and these we call heterotrophs. And we are a heterotroph, and we get most of our carbon from glucose or some molecule similar to glucose, meaning organic chemical molecule. Any question about that? So if you're ever confused about the classification of organisms based upon their energy source and their carbon source, just break it down to where do they get their energy source? If it's light, it's a phototroph. If it's a chemical, it's a chemotroph. And where do they get their carbon source? If they get it from CO2, they're an autotroph. And if they get it from an organic chemical molecule, they're a heterotroph. Because this is a very simple classification system. It gets a little more complicated when we consider both and then classify the organism on all four classifications. When we combine the energy source and the carbon source, we then have photoautotrophs, and they get their energy from light and their carbon from CO2. And we will mention there's oxygenic forms and anoxygenic forms. And I'm going to skip this slide and move on to the next. And I think everyone knows that green plants are photoautotrophs, but there are green bacteria like the cyanobacteria, which also get their energy source from light and their carbon source from CO2. So they're photoautotrophs. There are examples of oxygenic photoautotrophs that produce oxygen, both cyanobacteria and green plants. You notice how I'm saying green plants, because there are about a dozen plants which are not photosynthetic and they are not green. They're actually parasites, and they're not photoautotrophs, obviously. What would they be? If they're, I guess they're a chemoheterotroph if they're a parasite. So there are a few plants which are actually chemoheterotrophs, and they are not photosynthetic. They are parasitic plants, and they parasitize other plants. You don't need to know that, but I'm just mentioning that I'm using the word green plants. There are anoxygenic photoautotrophs, and the green and the purple sulfur bacteria are photoautotrophs, meaning they're photosynthetic using light and CO2 for their carbon source, and they don't produce oxygen. And I don't think anyone's ever worked with the green and the purple sulfur bacteria, so you don't really need to know that. Let's move down to the chemoheterotrophs. They're in the next easiest group to understand because we're a chemoheterotroph. They get their energy source from a chemical compound, And that could be something similar to glucose, meaning an organic chemical compound, and we're that. However, there are some prokaryotes that get their energy source from an inorganic chemical compound, and they are chemoheterotrophs. They are usually a chemosynthetic bacteria, and we'll talk a little bit more about that. The point is, instead of getting their energy needs from an organic molecule like glucose, they're getting it from an inorganic chemical compound, something like iron, hydrogen sulfide, or a nitrogen compound like nitrite or nitrate, meaning NO3 with a -2 charges. They get their carbon source, the chemoheterotrophs, from an organic chemical compound such as glucose. All animals are chemoheterotrophs, the protozoa, fungi, many bacteria like E. coli are chemoheterotrophs. Not all. The fermentative bacteria that make yogurt, for example, are chemoheterotrophs. Any question about the chemoheterotrophs? All right. The next easiest group to discuss are the chemoautotrophs, and they get their energy source from a chemical compound, but it's an inorganic chemical compound, such as iron, hydrogen sulfide gas, or nitrate, okay? And once again, it's the chemosynthetic bacteria, which are chemoautotrophs. And you'll notice that the chemosynthetic bacteria are split into two classifications. They can be chemoautotrophs or they can be chemoheterotrophs. The difference is where they get their CO2. The chemoautotrophs or chemosynthetic bacteria that use CO2 for their carbon source. The chemoheterotrophic chemosynthetic bacteria get their carbon source, from an organic chemical compound such as glucose. Okay. You're actually familiar with one chemoautotroph. It's a bacteria, like an iron oxidizing bacteria. And I bet you don't know where that you would see that bacteria, but everyone's seen it, at least everyone I know of. Okay. Anyone have a guess? When you take the lid off of the toilet tank, not the toilet bowl, but the toilet tank that stores the water, you see that yellowish or brownish sludge on the side of the toilet tank. That's actually iron oxidizing bacteria or some other nutrient oxidizing bacteria. That's a chemoheterotroph living in the water using CO2 as its carbon source and an inorganic chemical compound, which usually for the toilet tank is iron. Any question about any of that? All right. The fourth group, the hardest one to explain, are the photoheterotrophs. They obtain their energy source from light. That's why they're a phototroph. And they obtain their carbon source from an organic chemical compound such as glucose. There are some bacteria which are photoheterotrophs, such as the green and the purple non-sulfur bacteria. And who has ever heard of the green and purple non-sulfur bacteria? Anyone? Well, nobody's saying anything. My bet is no one's ever heard of the green and purple non-sulfur bacteria, but those are two types of bacteria which are photoheterotrophs. So in the chemoheterotrophs, oftentimes the energy source and the carbon source is the same molecule, such as glucose. And for us, we get both our energy source and our carbon source from molecules like glucose. But it doesn't have to be that way. Any question about any of that? What did I want to talk about? I think I don't want to talk about the lithotrophs here. So I think I'll just move on. You do need to know the nutritional classification of organisms that's in the red box. So we can start with all organisms. Ah, darn. Blow that up a little. Get that out of the way. Come on. We can divide organisms by their energy source. If they get their energy from a chemical compound, they're a chemotroph. If they get their energy from light, they're a phototroph. Of the chemotrophs, we can then divide them on their carbon source. If they get their carbon from an organic compound such as glucose, we call them chemoheterotrophs. If they get their carbon from CO2, they're called chemoautotrophs. For the phototrophs, we then divide them by their carbon source. And if they get their carbon from an organic compound such as glucose, they're photoheterotrophs. And if they get their carbon from CO2, they're photoautotrophs. You can further divide the photoautotrophs into the oxygenic ones that produce oxygen, and then the anoxygenic ones that do not produce oxygen. And you don't need to know that. It is true with the chemoheterotrophs, you can then divide them what their final electron acceptor is. And we'll talk about that when we talk about aerobic respiration. If the final electron acceptor, meaning where the electron ends up, is oxygen, then they are, well, the final electron acceptor is oxygen, and all animals are that way. Most fungi, protozoa, most bacteria also are that way. If the final electron acceptor is not oxygen, then they are what do you call that? An anaerobe. And you can split the anaerobes into whether they have an organic compound or an inorganic compound. An organic compound means the electron flows, like in fermentation, to an organic compound. And in anaerobic respiration, the electron flows to an inorganic compound, but it's not oxygen. And well, anyways, you don't need to know anything below the red box. Any question about any of that? So when we're talking about energy production, do realize that nutrient molecules have energy associated with them. And the energy is in the electrons that form the chemical bonds holding the molecule or the atom together. Well, you wouldn't hold the atom together. It's the chemical bonds between atoms that hold the molecule together. In cells, the energy is can be obtained from catabolic reactions, so that the energy stored in at least some of the bonds can be harvested by breaking the bond, and then that energy is used to make ATP. Realize animals are really poor at doing this, and we can only have 2 catabolic reactions to make ATP. I should say, yeah, that's true of all animals. And that's aerobic respiration and fermentation. And we'll talk a little bit more about both of those. I think it's in this lesson, actually. And then that energy is used to make ATP. ATP is called the energy carrier of cells because usually when cells need energy, they will take it by splitting a phosphate group or maybe even two off of ATP, and that will release energy from a high energy bond in ATP, and that energy is quick and easy to obtain. So if the cell needs energy, it usually gets it from ATP. And that's why we call ATP the energy currency in cells. Sort of like if you need to buy something, we use currency. Well, if the cell needs energy, it obtains that energy from ATP. Any question about any of that? So when we're talking about the production of energy, cells obtain energy from oxidation reduction reactions. Oxidation is when an atom or molecule loses an electron, and reduction is when an atom or when an atom or a molecule gains an electron. These reactions, oxidation and reduction, are always paired. So one entity, meaning one atom or molecule, will lose the electron, and another atom or molecule will gain the electron. Oxidation reduction reactions has an abbreviation. Apparently somebody decided it was too long of a name, and so they called it redox, which is actually reduction oxidation, but you get the idea. Okay, so redox stands for reduction and then an example of that would be when A loses an electron, and that electron moves to B. A becomes oxidized because it loses the electron. B becomes reduced because it gains the electron. An exact example of that is when copper and iron +3, and I guess copper +1, reacts with each other. I wonder why the arrow isn't showing there. And then copper loses an electron and becomes copper 2+, meaning 2 positive charges. The iron gains an electron and then changes from iron 3+ to iron 2+. Any question about that? An easy way to remember oxidation and reduction is to say the mnemonic, whatever you say that. Leo the lion goes grr. Leo, lose electrons, oxidation. So L for lose, E for electrons, O oxidation. Leo, the lion, goes grr, grr, G for gain, E for electron, R for reduction. So the molecule that loses the electron, Leo, the lion goes grr, the molecule that gains the electron becomes reduced. Any question about any of that? So we use oxidation reduction reactions to make ATP and actually all other molecules we make. In biological systems, oxidation usually has a proton, meaning H+ ion, removed with the electron. And then the H+ ion usually follows the electron. So then in biological oxidations, we often call those dehydrogenations because besides losing an electron, this molecule, which is going to become oxidized, is going to also lose a hydrogen ion. And then the same thing with the molecule which is reduced. Besides gaining an electron, it's going to gain a hydrogen ion. Let me see if I can blow that up a little. And remember that a hydrogen atom is the hydrogen ion and then the electron. So we'll actually talk about NAD plus, a coenzyme, which we may have talked about already, gains the electron and the hydrogen, and actually comes from glucose, or a molecule derived from glucose. And then the NAD plus becomes reduced by gaining the electron, and it gains the hydrogen ion as well. Let me see, where's the hydrogen ion? Right here. And there it is there. That's the extra hydrogen ion. That's the extra electron. And NAD plus actually gains 2 electrons, which you don't need to know. That's shown there. And then the molecule, which is either glucose or derived from glucose, becomes oxidized because it loses a electron. Yes. Any question about that? All right. So biological oxidation reduction reactions that we call redox reactions are used in catabolism to extract energy from nutrient molecules. So when we eat sugar, we can break down sugar to obtain energy and use some of that energy to make ATP. We only use highly reduced compounds. A highly reduced compound is one that has many hydrogen atoms, can be degraded, meaning catabolized, to a highly oxidized compound like CO2 and water to produce energy. And like I said, our cells are really poor at doing this. We can only do this making ATP from glucose or a molecule similar to that in the case of aerobic respiration or in the case of fermentation. All other catabolic reactions, even using glucose, would not generate ATP. Like sucrose, when we split sucrose into glucose and fructose, we cannot generate ATP from that catabolic reaction. The highly reduced compounds like glucose have a lot of energy associated with them, and that's a lot of potential energy, which is stored in the chemical bonds of the molecule. So let's move on and start talking about aerobic respiration now. Aerobic respiration is how cells oftentimes make ATP. So they convert the energy in a molecule like glucose, and then they will store a lot of that energy that was in the molecule of glucose in ATP. Aerobic respiration specifically uses glucose and oxygen going through a couple of steps in either aerobic respiration or cellular respiration. And that term, cellular respiration, is used interchangeably with aerobic respiration. And I'm not sure why that came about, because cellular respiration technically only means respiration in a cell, and the cell can perform, at least some cells can perform anaerobic respiration. Okay. But if it's anaerobic respiration, it's not cellular respiration, because cellular respiration is used synonymously with aerobic respiration. So I'll try not to use the term cellular respiration. But in the cellular respiration, some of that energy that was stored in glucose is converted into heat, and that's just the second law of thermodynamics once again. Some of that energy released by the glucose is stored in the energy of ATP, and it's used to make ATP. And then the glucose and the oxygen are converted into carbon dioxide and water. The cell will then use the ATP to do cellular work, meaning whenever the cell needs energy to get something done, it'll obtain that energy from ATP. Any question about any of that? All right, here's a summary reaction for aerobic respiration. Glucose, and you should probably know this, C6H12O6, plus 6 oxygen molecules, plus 38 ADP, adenosine diphosphate, plus 38 phosphates, and the I just stands for inorganic phosphate, derive or make 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. This is the overall summary equation for aerobic respiration. Any question about any of that? Now, in reality, aerobic respiration doesn't happen in one step. It happens in a number of steps, but this is the summary equation for aerobic respiration. So in the generation of ATP, some of the energy from some of the redox reactions is used by the cell to make ATP. And not all oxidation reduction reactions can make ATP in animal cells, only two, aerobic respiration and fermentation. And then some of the energy that comes out of glucose can be converted and stored in the energy stored in ATP. Obviously, some of that energy is lost as heat. When we're making ATP, ATP can be made, well, I guess I'm not talking about that yet. ATP, the making of ATP is by a phosphorylation reaction. And what happens is ADP is phosphorylated. Phosphorylation is simply the addition of a phosphate group. So ADP, that's adenosine diphosphate, meaning two phosphate groups. You need to add energy for this reaction. And then you add phosphate. And then you can make, which is the arrow here, ATP, which is adenosine triphosphate. Let me blow that up. You'll note that ATP has two chemical bonds, which are high energy bonds, the third phosphate and the second phosphate. ADP has one high energy bond, the second phosphate group. And when the cell wants energy, it can break this chemical bond and then use that energy to do whatever the cell wants to do that requires energy. We call that cellular work. If the cell wants more energy, it can then break this chemical bond from ATP. So it breaks both of them. And it can obtain the energy from that bond and then also that bond. Any question about any of that? Cells can make ATP by three different mechanisms. You can make ATP by substrate level phosphorylation, where you have one molecule of ADP and then another molecule which has a phosphate group on it, and then transfer this phosphate group directly onto ADP to make ATP. And then the molecule which had the phosphate group becomes the molecule without the phosphate group. This substrate level phosphorylation does happen in fermentation and glycolysis, but we're not really going to talk much about it. But this is the first way that cells can make ATP. The second way that cells can make ATP is by respiration. And it does have another name. You could call it oxidative phosphorylation. Respiration involves a sequential transfer of electrons from an electron donor, where the electron was derived from, to electron acceptors, and there's going to be many of them. And then they'll transfer the electron to another molecule, transfer to another, 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 and then finally transfer to the last electron acceptor. We call that the final electron acceptor. In an aerobic respiration, the final electron acceptor is oxygen that receives the electron. The point is, in respiration, energy is released along the process, and then ATP can be generated by a process called chemoosmosis, and we'll talk about that in just a few slides. The point is that the second way that cells can make ATP is by respiration. In our cells, we do it by aerobic respiration, but respiration can happen anaerobically in some cells, particularly anaerobic bacteria or anaerobic archaea. No animal cells can make it by anaerobic respiration, but bacteria can. And it's a process, all of it's always similar to this here, which you don't need to know the process. You just need to know. The second way cells can make ATP is by respiration. The third way that cells can make ATP is by photophosphorylation. Photophosphorylation only occurs in photosynthetic organisms. What happens is light releases electrons from chlorophyll, and then the light energy via the electrons are converted into chemical energy via an electron transport chain. And ATP do get made in photosynthesis or photophosphorylation. However, oftentimes in photosynthesis, the ATP and the NADPH that are made are often used up to make sugar, so that the light energy is initially converted into the energy in ATP, but then the energy in ATP is used up, so ATP is broken down into ADP again to make glucose. Okay? And this picture is showing you an example of photophosphorylation. The green plants, obviously, and these very muddy boys, which must be photosynthetic because they're all covered in mud. That's a joke there. Any question about any of that? All right. So the energy that's released from molecules is controlled and stored by a series of reactions. So when we release the energy in glucose to make ATP, it doesn't happen in a single reaction, in a single release of the energy. And if you were to release the energy all at once, you would, like if I had a beaker of glucose here, and then I would break it down by taking a blowtorch to it and catching the glucose on fire, there would be so much energy released all at once that would kill the cell. So the cells don't release it in one step. They release it in a series of steps. And the electrons from the donor molecules are passed from one compound to another through a series of redox reactions. And I actually showed you that happening here. It doesn't happen at one step. Actually, it happens right before making the NADH, and then here, and then here, and here, and here, and here, and here, and here, and then finally here. Any questions about that? All right. So many organisms, if not most organisms, can get energy from catabolism, and they use this as their primary source for cellular energy. The most common energy source is glucose, and the organism will break down glucose to make ATP using a catabolic reaction. But organisms don't have to use glucose. We can break down starch, and then, as you know, starch is just broken down into glucose, and so you send that the same way. But organisms may use lipids and proteins to obtain their energy needs to make ATP. Like marathon runners can burn lipids, fats, and other lipids to make ATP. However, a marathon runner doesn't burn lipids until 45 minutes into their run. And before that, the marathon runner is getting their energy from glucose or another carbohydrate, which will be broken down into glucose. Any question about any of that? All right, so let's talk about making ATP from glucose, and that does involve catabolism. Like I said, most organisms can do that. There are a few that do not, and you might think the green plants do not, but actually at nighttime, when a green plant needs energy, it will get its energy from breaking down glucose in aerobic respiration. So most organisms can get the energy they need to make ATP from catabolism of glucose. There are two processes that microorganisms use to make ATP: respiration and fermentation. So organisms can break down glucose by either respiration or fermentation to make ATP. When we're talking about respiration, there are two forms of respiration. There's aerobic respiration, where the electron will flow to oxygen, and that's the way people and all animals get most of their ATP. We can actually get some ATP from fermentation. We'll talk about that. And then some organisms can engage in anaerobic respiration, and that would be all anaerobic bacteria. And they can break down glucose to make ATP using anaerobic respiration. Are there any questions about any of that? If not, let's watch a little video on aerobic respiration. Don't have the sound there, but it looks like it's going to be a commercial. So let's turn on the sound now.
And CV. Grammar.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
This video is for Paper 1 of GCSE Biology and...
All right, any questions about that video? Not hearing anything, so I'm going to assume not. Let me stop my share to make sure there's people here. Yep, there are. Got a little worried there. All right, so this slide is showing you aerobic respiration and fermentation. We'll talk about fermentation after aerobic respiration. Aerobic respiration has 3 stages: glycolysis, the preparatory step in the Krebs cycle, and then the electron transport chain. Essentially, in aerobic respiration, the electrons flow from glucose to CO2 and water, and we make a lot of ATP. In fermentation, we make much less ATP. This slide is showing you an overview of aerobic respiration in a eukaryote cell. Only eukaryotes have mitochondria, but in a eukaryote cell, aerobic respiration starts with glycolysis, which occurs in the cytoplasm of the cell. So there's the cell, the box, glycolysis in the cytoplasm, and then some ATP is made. We'll talk about NADH in a little bit, and then the derivatives of glucose move into the matrix of the mitochondria, where we have the citric acid cycle, which is also the Krebs cycle. It makes some ATP. Come on, mouse. And we have more NADH and FADH2, and they go to the electron transport chain, which happens in and along the inner mitochondrial membrane. And here we make a lot of ATP. Now, in a prokaryote, that doesn't happen in the mitochondria. Glycolysis does happen in the cytoplasm, but the electron transport chain is actually in the cell membrane. So if this is the cell membrane, this is where the electron transport chain happens. All right, glycolysis is the oxidation of glucose to pyruvic acid. It does have another name, which I don't use. Glycolysis occurs in most living cells. It can occur in the presence or the absence of oxygen, and it does produce ATP and NADH. Here we're looking at the steps of glycolysis. Oops, that's in the way there. And it's in the way there too. We're going to have to just start. So you start with glucose, a 6-carbon molecule. And then you go through a number of steps. You add in 2 ATP, but you get out 4 ATP. And you end up with pyruvic acid. You do not need to know the steps of glycolysis, but you do need to know that you start with glucose. I need to add that on here. And then you get a net gain from glycolysis of 2 ATP molecules, 2 pyruvates, and we're going to call that the same as pyruvic acid. The cell uses these 2 molecules the same. Chemically, they are slightly different, but the cell uses them the same, so I'll use them interchangeably. And then you get 2 NADH from glycolysis. Any question about that? So you need to know the net chemical reaction of glycolysis. You don't need to know each of the steps. And then we have the, I guess I need to talk a little bit about respiration that occurs. It can happen from in aerobic respiration, where the electrons will flow to oxygen. So the final electron acceptor is oxygen. And then another way is anaerobic respiration, which some prokaryotes have, where the final electron acceptor is not oxygen, it's some other molecule other than oxygen, like a nitrogen compound or a sulfate compound. It depends on what form of respiration the cell is using, where that electron flows to. The point is, in respiration, ATP is generated by oxidative phosphorylation. We'll talk a little bit more about that in the next couple of slides. So the pyruvic acid cannot enter the Krebs cycle directly. And what the pyruvic acid does in one step, it's changed into acetyl coenzyme A. And we call this step the preparatory step, preparing the pyruvic acid for entry into the Krebs cycle. Pyruvic acid is a 3-carbon molecule. Acetyl coenzyme A is a 2-carbon molecule. So 1 carbon comes off and is burnt as CO2. This is the first CO2 that comes off of glucose. We do generate 1 NADH in the preparatory step. And we'll talk more about the NADH. Now, the preparatory step runs 2 times per molecule of glucose because in glycolysis, from 1 glucose, we get 2 pyruvates. So the preparatory step will run twice for each molecule of glucose. All right, the acetyl coenzyme A can now enter the Krebs cycle. The Krebs cycle does have a number of other names. It's called Krebs cycle after the doctor who discovered it, Dr. Krebs. It's also called the tricarboxylic acid cycle because it has a bunch of tricarboxylic acids in it. It's also called the citric acid cycle because that's the first tricarboxylic acid in the cycle. So it has a bunch of names, and sometimes it's called TCA for an abbreviation. Okay, I'm generally going to just call it the Krebs cycle, and your textbook author generally calls it the Krebs cycle also. So there's the preparatory step being shown here. The acetyl coenzyme A binds with oxaloacetic acid. It's converted into this molecule here, converted to that molecule here, converted here, converted there, converted here, converted here, converted there, converted back to oxaloacetic acid, so that we end up at the molecule we started with, and that's why it's called the cycle. And it will always run as long as acetyl coenzyme A is fed into the cycle. Any questions about any of that? All right, we do generate in the preparatory step in the Krebs cycle 4 NADH, 1 in the preparatory step, 3 in the Krebs cycle. So 1, 2, 3 NADH in the Krebs cycle. We do generate 1 FADH2, which is similar to NADH. They carry the electrons to the electron transport chain. There's the FADH2 here. We do generate ATP at this step of the Krebs cycle. The NADH and the FADH2 carry the electrons to the electron transport chain. And the preparatory step in the Krebs cycle will run 2 times for each molecule of glucose. Any question about any of that? All right, I'm not hearing anything, so let me go to the next slide. This is showing you an overview of the glycolysis happening here. And then we have the Krebs cycle shown here. And then the NADH and the FADH2 take the electrons either from glycolysis or the Krebs cycle to the electron transport chain. Any question about that? In the electron transport chain, we now make a lot of ATP. The electron transport chain, abbreviated often ETC, is a series of carrier molecules that are in turn reduced and oxidized as the electrons flow from glucose to, well, eventually they'll flow to oxygen, but the electrons flow from one molecule to another. And the electrons in the Krebs cycle and glycolysis flow to NADH or FADH2, and then they carry it to the electron transport chain. All right, any questions about any of that? So in eukaryotes, the electron transport chain is a bunch of proteins in a membrane, and this membrane is the inner mitochondrial membrane. In prokaryotes, it doesn't happen in the inner mitochondrial membrane because they don't have mitochondria. It happens in the cell membrane. So these proteins are proteins in a membrane, and it's a cell membrane. Any questions about any of that? All right. So the NADH and the FADH2 bring the electrons to these proteins in the membrane. And I should mention that that's why we call it the electron transport chain. The electrons are transported to these proteins, and then they transport from one protein to another. Come on, mouse. In the membrane. And that's why it's called the electron transport chain. It's a chain of proteins in a membrane. And the electron moves from one protein in the chain to another. NADH brings the electrons to the first protein in the membrane, meaning the first protein in the chain. FADH does not. It brings it to the second protein in the chain of proteins. Come on, mouse. Ah, there it is there. When the electrons flow through some of these proteins, the electron gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. And that happens with this protein, with that protein, and with that protein. This creates a twofold hydrogen ion gradient, meaning there will be more hydrogen ions on this side of the membrane than on this side. There will also be more positive charges on this side of the membrane than that side of the membrane. And that creates a twofold gradient difference where these hydrogen ions will want to move from this side of the membrane to this side of the membrane. However, to do that, the proteins can only move through this protein channel here. They cannot move through the bilayer of the membrane, but they can move through this channel protein. This channel protein is ATP synthase, and as a hydrogen ion moves through it, the energy of the hydrogen ion moving through it gives the protein the energy it needs to phosphorylate ADP to make ATP. And that's how cells make ATP. They pump hydrogen ions from this side of the membrane to that side of the membrane. That hydrogen ion gradient is a bunch of potential energy. The cells use that potential energy to then make ATP. Any question about any of that? All right. There's one other reaction that happens, and that is as the electron flows to this, through this protein, to this protein, to this protein, to this protein, to that protein, it then leaves the chain of proteins and goes to the final electron acceptor. In aerobic respiration, this electron flows to oxygen, shown here. Come on, mouse. The oxygen combines with electrons and then two hydrogen ions to make water. So in aerobic respiration, besides making ATP, we do make, in the electron transport chain, we do make water. The water may be breathed out in people. The cell can use it for its own water needs, and actually desert animals get something like 60% of the water they need from aerobic respiration. And that's because they're very good at conserving this water, and they then use it for their water needs. Any question about any of that? All right. What else did I want to talk about? Oh, yeah. I need to say that NADH is a little more efficient at making ATP because the electrons flow through three proteins that can pump hydrogen ions from this side of the membrane to that. And FADH2, the electrons only flow through two of the proteins that can pump hydrogen ions across the membrane. So in the electron transport chain of aerobic respiration, in prokaryotes, which are a little more efficient, they can generate up to 34 ATP molecules per molecule of glucose anaerobic respiration. Eukaryotes are not as efficient. Most cells can only generate 32 ATP molecules per molecule of glucose, but the heart, the liver, and the kidneys can generate 34 molecules of ATP per molecule of glucose. And you don't need to know why, but in the heart and the liver and the kidneys, the ATP, excuse me, not the ATP, the NADH coming from the cytoplasm in glycolysis stays as NADH. And let me blow this up. And so it goes through 3 proteins that pump hydrogen ions through it. And all other cells of the body, meaning not the heart, not the liver, not the kidney, such as your skin, your nose, your brain, that NADH is converted to FADH2, which is not as efficient. So most of the cells in our body generate 32 molecules of ATP per molecule of glucose. But the heart, the liver, and the kidney are more efficient, and they can generate 34 molecules of ATP. Why do you think the heart, the liver, and the kidney are more efficient? What do you know about the heart, the liver, and the kidney? Or what do you know about the heart? Come on, you guys obviously know one thing about the heart.
It's more mitochondria.
That's not entirely true. Like your muscles, for example, if you're really working out, you're going to have a lot of mitochondria, and the muscles only generate 32 molecules of ATP. But that's a good guess. What else do you know about your heart muscle compared to, say, your muscles, your arm or something?
Is it because it's thicker?
The heart? I don't know the answer to that. No, what I'm getting at is the heart, the liver, and the kidney, they're always working. They're always working, so they're always needing energy. And that's why they're more efficient than the cells of your skin or even the cells of your muscles. They're not always working, so they don't need to be as efficient as the heart, the liver, and the kidney. Any question about any of that? All right. I think I've already talked about that, and I've talked about that, and I think I've talked about that too. Let's go ahead and blow that up. In the Krebs cycle, we mentioned that the electron releases that energy in steps, and then that energy is used as the NADH or the FADH2 delivers the electrons here. This is one thing I want to say. And as I did say, let me blow this up. In eukaryotes, this happens in the mitochondria. At least the Krebs cycle is in the matrix of the mitochondria, which is here or here. And then the electron transport chain happens in and across the inner membrane of the mitochondria. Now, in bacteria, they don't have a mitochondria, and that happens in the cell membrane. And I guess I should mention that the hydrogen ions in a eukaryote are pumped in this space between the outer membrane and the inner membrane. So this is one way that our mitochondria protect their cell. These hydrogen ions aren't going to come out and muck other places of the cell. In the prokaryote, of course, it protects the cell because the hydrogen ion is actually pumped outside of the cell. And these hydrogen ions are actually pumped into the cell wall. Come on, mouse. There we go. And then the hydrogen ions flow through this channel protein, which is ATP synthase. All right. that I think is it for aerobic respiration. We already talked about that. The movement of hydrogen ions is called chemiosmosis. I don't tend to use that term, but that is a term. And then the movement of hydrogen ions is used to make ATP. And I forgot to mention that the electron transport chain has a large number of names. It's sometimes called electron transport phosphorylation because ATP is, or ADP is phosphorylated to make ATP. It's also called oxidative phosphorylation. It's a term I don't use. I generally use, and your textbook uses, electron transport chain, and we may abbreviate it ETC. Okay. It has another name, electron transport chain, or it's sometimes called electron transfer chain. All right, so that means the complete oxidation of glucose via aerobic respiration, counting the 2 ATP made in glycolysis, the 2 ATP made in the Krebs cycle, and then the ATP made in the electron transport chain, means that of eukaryotes, we get 36 to 38 ATP molecules per molecule of glucose. And the heart and the liver and the kidneys are the only cells that can make 38. All of our other cells make 36. Prokaryotes are a little more efficient, and they generate 38 ATP per molecule of glucose. And that gives us our summary equation for aerobic respiration in prokaryotes, where we have C6H12O6 glucose plus 6 oxygen molecules plus 38 ADP, 38 phosphates, generate 6 CO2, 6 water molecules, and 38 ATP molecules, which is shown here. Why is that not showing? There we go. All right, any question about any of that? If not, I think I'll stop here. I think we're at our ending time, and I'll see you, I believe it's 6:30 for the lab. Any questions? All right, I'll see you in 6:30 for the lab.